It's been a fantastic morning listening about different systems at different scales. I think we've been able to understand now of looking at small measurements and how they can be put into a model to be interacted with, and then also how we've just seen some very large scale models and how these can be brought forward. So I think one of the immediate questions I thought about, which was put up there by Greg, was that how do we actually evaluate these tipping point hypotheses? And I think that crosses all of these different talks that we saw and all these different approaches, whether it's a systems biology approach or whether it's other approaches. So I was wondering if, Greg, do you want to just start off on that? Now, don't forget, this is also like a dating component here as well. Everyone's going to look at each other a bit stunned at the start, but you know, these are nice guys. I can vouch for them. So please ask their questions. So, Greg?
Yeah, so I think that's the fundamental question. And unfortunately, I don't know enough about your communities to say anything intelligent, aside from the fact that the scientific method really is about making predictions and testing hypotheses. And in the modern era, that really involves numerical models and sophisticated measurements. And when those two disagree, we have to correct the models, so we need better measurements. And using these state estimation techniques can be one way forward to try and bring them closer together to figure out where the problems are.
So on that question as well, I suppose one of the things that when you start to make these models and you're thinking about testing them and trying to evaluate them, how do you actually evaluate scale as well? Are they scalable?
Yeah, that's a good question. So let me give you an example. So one of the things we're working with for this climate estimation problem, we're trying to do paleoclimate reconstruction, where we're dealing with thousands of years and proxy measurements. And unfortunately, we can't do this ensemble sampling technique with these what are called kitchen sink complexified models. They're too expensive to run an ensemble for 1,000 years. But it turns out we can estimate these models empirically using what's called linear inverse modeling. So we effectively try and find low-dimensional versions of these models, and they're empirically estimated in some measure that provide forecasts. And the interesting thing is, their forecasts are pretty darn good. When you compare the forecasts from these simple linear models against the kitchen sink models on, say, a 5 or 10 year time scale, they're pretty good. If you have enough data about your system and it's not too strongly nonlinear, there are ways to estimate these models empirically.
Dustin, I think you're probably next in the scale of the panelists here, looking at these broad species and the way that they're interacting with the environment. How does that types of approaches work with scaling for ornithology?
Well, we run into some of the data issues. Also, our data are a lot cruder than some of the experimental systems. Our measurements, when we look at physiology, are not as precise as we heard yesterday with some of the things. And so, and then when we work in wild organisms, we have long generation times. So for the model that I talked about at the end, the obvious place to start testing those tipping points is in yeast systems, in plankton systems, in biofilm systems, to test critically what happens to these populations as you approach these boundaries and go towards tipping points. Now, when you take that back into the field, we're not there yet, I think, about how we're going to do it, largely because the populations take so long to breed. And what I guess I would say we don't want to get to the climate modeling approach and say, okay, well, these populations have gone extinct, and so what can we learn from that? We'd like to avoid that. And so there's some happy medium. There'll probably be some populations in the wild that we can study that have shorter generation times. I don't know, maybe things like Drosophila that we think about as a lab organism, but that you can do in the lab. And I said that's going to be the critical area to focus on, I think.
How do you deal with intelligence, though? Because that's another dimension here. Because with environment and climate, that there's no intelligence really innate there, other than other impacts. But with your systems, you've got immediate intelligence making decisions as well.
You mean you mean in terms of policy, how to protect?
Well, just that, if you've got those species of birds that are having to decide whether to go fly left or fly right, that also has an impact.
Yeah. So, yes. So in terms of, for certain things, like, it gets dicey when you start thinking about behavioral things like this, right? So behavior is a much more, I would say, plastic trait in general. It's more labile. It can flip on and flip off on the evolutionary tree. So if you look at cooperative breeding, going back over the evolutionary history of birds, it probably evolved early on, and it's been lost and gained in lineages. And so I tend to shy away from saying, people ask, well, are species that are not social now going to adapt by becoming social? That would be one prediction you might make, and I shy away from that. We just don't know. But in theory, a lot of birds occasionally do show this behavior. You get reports from birders all over the world where they see a weird observation of more than two individuals, more than a pair raising offspring. So maybe that is a possibility down the road.
Before I dominate, next.
So with your question with respect to optimal sensor placement, it occurs to me that with biological networks, large numbers of possible things to measure in a particular system to determine things, you'd want to hone in on the measurements that make the most sense. Has there anything been done in terms of that for a network, like a biological network system?
Although this idea has been around for quite a long time, I think doing it robustly is a relatively recent development, that is, accounting for the uncertainty. Really, the most important thing is knowing what you want out of the measurements. And I think what you're getting at is it may be hard to know, especially if you're interested in something that's connected to many other things. So just to make it specific for our applications, if NSF is interested in monitoring temperature at South Pole Station because they can't get the planes off the ground if it's too cold, that's very, very specific. We can optimize for that. If you're interested in, say, the health of a forest, depending on lots of different things, including precipitation, but also an infestation that may affect the trees. In other words, it could depend on lots of things. I think it's a much harder problem.
And I guess sort of semi-related question in terms of, do climate models yet include biological feedback loops, for example, bacteria?
Absolutely. Okay. Yeah, absolutely. And they have important global teleconnections. Yeah.
Sui?
Yeah, so given the diversity of the panel here, I have a very broad and philosophical question. And that is, we heard a lot about predictive models. We do that a lot with predictive medicine. But now we just heard a talk about forecast, right? Like weather forecast, climate forecast. And for me, this is I don't know whether they are the same thing or not, but we heard Nitin's talk and Dustin and Steve's talk about predicting relationships, predicting a pathway, the existence of a biochemical pathway. Or in chemistry, we have a theory of the atom, and we predict the existence of elements that you have never seen. That's slightly different, I think, from forecasting, giving a dynamical system, which is knowing the structure or not, as we heard in the first talk by George Tsukihara. Without the structure, you try to forecast the temporal evolution of a system. And I wonder whether there is a connection between these two and how people think about that. And maybe George can also comment on that.
You don't want to overpopulate your model with variables, but if you have to understand your system to choose those variables, I think. And one of the things that's often missing from these conversations is the things that you don't know that you need to know are going to come as a surprise. And so that's why it says on the opening page of the National Science Foundation where discovery begins. So how you improve your success rate at filling in those parts of the knowledge that you need to complete your model. I'm, of course, thinking of the oceans. I don't know how to solve that, but until that more information comes in, I don't think we can make a very complete model for the biological carbon pump.
So I can comment on the models we build. Even in the microbial systems, this data space becomes so large when you query the behaviors across many different environments that the chance of finding coincidental correlations also rises. So the question of how do you constrain the modeling to what the correlations that are meaningful is something that we really pay close attention to. And one way we do it is by grounding the models in known biological mechanisms of how things work, but leaving room for discovery at the same time. For example, when you look at gene regulation, we know certain ways that gene regulation occurs, and we can look for evidence of those patterns to support some of the correlations we find. But when we investigate the whole system, we also leave room for finding some new patterns that may arise that we had not previously anticipated. But in the end, when we get these models that explain the data that we've generated, we also make predictions about new interactions, maybe regulatory interactions that we can go back in the laboratory and test. And to us, that iteration is really important to then get closer to the mechanistic basis for the correlation so we can get to cause and effect.
Before you jump, can I just add a point that I think independent verification is really important, either by having independent measurements of the forecast or ideally having some sort of double-blind mechanism. Now, this is not the case in the climate world. And it's interesting that we have all these different climate models, and they reproduce the 20th century global mean air temperature all very, very well. They're all very similar. And as soon as you go off into the future, this happens, right? And I think that's because they've all been tuned to the same thing. They're all targeting the same thing. If we had some double-blind mechanism where these models were going to be evaluated against things you don't know about, it would take away some of this fiat tuning that's taken place. So that's an idea I think worth considering.
George.
So I think there's a really, you just raised a really important distinction between fitting versus predicting. And I mean, what you just said sort of makes me uncomfortable about long-term climate prediction, right? Especially with, if you have very large positive Lyapunov exponents in the dynamics, then, you have this great uncertainty about going into the future. But one thing that you said that I thought was really interesting was that if you linearize the system, so for example, maybe think of it as an AR, an autoregressive process, and you create this very, very simple model, you can actually track things reasonably well, probably in the very short term, for very short-term forecasts. This is a pretty common problem in a lot of different disciplines. There's something called the FERBIS model, Federal Reserve of the United States model, which is probably not quite as complicated as your climate models, but they have on the order of 25,000 to 30,000 parameters. And it's a model that I think basically is can be used to justify any policy you want to create, but it does not pass the test of real prediction, of real forecasting. In fact, if you make a simple AR2, you can actually do better. So a very simple linear autoregressive model. But then that linear autoregressive model does not contain much understanding, right? And so there's a trade-off here between what you want to be able to do is predict discover what the important driving causal factors are, or what the really relevant causal factors are, and then test that set of discovered things with a reasonable forecast. But I think ultimately forecasting, out-of-sample forecasting, not fitting, should be the ultimate test of the validity of a model, of our understanding.
I guess I would say,
Sorry?
as an ecologist, I often think, compared to, say, a physicist, our data are sloppy, right? So I once had a conversation with a physicist, and think about regression. He fit a regression line, and he had a R squared of 0.99 and was very happy. And if he got 0.96, he was very unhappy. If I get 0.6, I'm very happy. But then he says, you can't predict, you have no predictive power from your regression. And I said, I don't really want to predict. I want to explain the pattern in my data. I don't necessarily need to predict out, whereas the physicist wanted to predict out further from his things. And so it depends, I guess, on the way we approach it. And ideally, we want more variables and better data, but that's not always the reality in some of our systems.
So there's a bit of tension on this topic, I think, in the weather and climate modeling world, that these are really complicated models. They're 100,000 plus lines of code. They have all these different components to them, right? And you have researcher X goes into the field to measure quantity Y, and they're going to constrain this parameter that lies in this model, right? So they've done this first principle science, and they could tell you in the field, in nature, this is what the value of this parameter is, and here's its range of uncertainty. And expects to put that into the model and improve predictions. And most of the time it doesn't, because it's conditional upon 5,000 other parameters in that model. So this really is a fundamental question. Would you rather make a good forecast that has value for humanity or for the Fed or whoever, or would you rather really understand all the components and not how they fit together?
Yeah, but that understanding is basically a hypothesis.
It is, absolutely, yeah.
So I don't know. I think that
George, do you want to use the mic? Because the people at the back don't hear.
so. Essentially, any model is just a hypothesis, and it seems like if you want to understand the validity of the hypothesis, that forecasting is the way to do it. I feel a little bit uncomfortable that you can do so well with a linear model, but not as well with the full with the full nonlinear model. And so to me, that would suggest that maybe the model is not. Maybe there's some problem with with our ideas about how we actually put all these little pieces together. So I think the individual pieces are totally legitimate. I mean, there's no doubt about it. But then we make some decision about how those pieces about the functional form, basically. So, there's a paper actually that one of my students just came out in PNAS last week on predicting salmon in Fraser River, Canada, not far from here. And it turns out that if the conventional models don't do very well, and if you include temperature, which everyone knows is important, they do worse. I think what that does is it kind of underscores the fact that these models are really just our idea, are just basically a hypothesis of what we think the world should look like. But and what we really need to do is examine. This should be evidence-based science. We should examine how well this model actually does in a real-time forecast, which is the real measure of validity.
Larry.
Yeah, Larry Smar, UC San Diego. So I wanted to crisscross a couple of the panelists between the climate and the ocean microbial world, since most of the planet's life is microbial. And I remember when Ginger Armbrust told me, I think 20% of the oxygen in the atmosphere comes from the diatoms. The changes in tipping points that couple the climate models with the microbial sensitivities that we heard about to changes in the local variables. Now, MIT has Project Darwin, where they've worked for a number of years on doing this. So in other words, they have maps of the whole ocean in which you get the concentration of many different species of microbes as the underlying variables change in the climate model. But what I worry about is tipping points that are bio-climate coupled and something you wouldn't necessarily see separately, that is in the non-microbial climate or just the local microbial studies that were beautifully described. So, discuss that.
Steve, are you
Everybody's looking at me. Yeah, that's an interesting thought, Larry. I'm not sure how to answer. Those microbial communities are extremely complex, but what part of that complexity do you really need to know about to build a model? So, I mean, let's take the discovery of Animox Okay, you needed to know about that to build your model, right? To build models for nitrogen, at least for nitrogen cycling in the ocean. There was a big piece of the puzzle. Nothing was known about it. Then these bacteria are discovered. Now you can understand that and add it to your model. So what other things like that are out there? And I think that's partly what you're saying. Does it matter? I'll take from my talk, there are lots of different, not too many, but a dozen, less than 100 important groups of bacteria that turn that carbon back to CO2. In your model, should that be 1 box, 4 boxes, 15 boxes? So I don't think we have enough information to make those kinds of decisions right now. We don't know what most of them do, except that they are not photosynthetic, so they must do something else.
Hi, I'm Sarah DeWeert. I'm a freelance journalist, and I have a question that I guess is first for Nitin, and then maybe a follow-up that might also involve Dustin's work. So this idea that relaxing constraints can broaden niches and potentially enable some organisms to kind of boot out others and outcompete others. So we've seen that in the microbial world in the oceans, but I'm wondering to what degree do we think that that is kind of a general property of organisms and a potential for sort of, we might see it more broadly with climate change. And then I guess the part that might involve Dustin's work is how might that dynamic interact with your sort of model of tipping points in this sort of evolutionary adaptive space. I guess the overall question being, how do we sort of predict who the winners and losers are going to be?
I'll try to answer your question in terms of the applicability to what we find in the microbial world to the larger organisms. I think there is evidence that when you have an exotic species that is introduced to a habitat where it doesn't have predators, then it can take over and essentially change the dynamics of interactions in the resident populations there. And even with the increasing temperatures, you see tree growth, for example, kind of going up the mountains and so on, and just changing the dynamics of the ecology or the ecological interactions there. So I think it's a fairly general phenomenon, and maybe Dustin can comment on that more.
Yeah, I'll give you 2 examples from other systems that we study sort of related to this. So I also study social invertebrates. So we study burying beetles a little bit, and burying beetles can cooperate and not cooperate. And if you look at beetle cooperation over an environmental gradient, say up a mountain, those that are able to cooperate are able to extend their environmental niche, and by cooperating, they're able to outcompete competitors. So burying beetles lay their larvae on carcasses and have to compete with fly larvae. And so by being cooperative, they're able to outcompete competitors and extend their environmental niche. We also study eusocial snapping shrimp, so in the marine environment, and these are symbiotes with sponges mainly. And what we know is that So they range from non-social species that live in pairs all the way to eusocial colonies that live like termites, bees, and wasps, and everything in between within a small group of about 45 species in a recent radiation. And we know that the social species are actually able to outcompete the non-social species. They have a wider niche. They can inhabit more sponges and do better that way. But what we're finding is actually they may be more susceptible to these tipping points. So in three islands in the Caribbean where we have long-term data, the one island in Belize where the eusocial species were most abundant on the reef, the most diversity, they've gone locally extinct in the last 5 years. We don't yet know why, and we're seeing similar trends in other islands. And so I think that illustrates how there are these behavioral adaptations that allow them to outcompete, but maybe they actually are more susceptible to environmental perturbations. We don't know what the perturbations are, but it's probably water temperature change, salinity change, or or sponge disease that has taken their primary host. And so I'm not sure what to predict. It's going to depend on the type of adaptation, probably, whether it's behavioral, whether it's physiological, whether it's genetic, and it's going to vary by species and perhaps in these different environments also.
Next question.
So my question is sort of a reflection on Steve's observation of genomic plasticity in the microorganisms in response to their environment. So would you say it's fair to say that maybe genome should be considered as a particular structure of genome in a given cell should be considered as sort of a result of dynamic attractor or dynamic equilibrium, where not only the genome determines biochemistry, but biochemistry that reacts to environment in terms maintains through a set of feedbacks, maintains the genomic structure. And perhaps in some circumstances, you can even see the phase transition in genome and complete rearrangement.
I think I completely agree. Yeah, I mean, that sounds like a description of the way I view it. I mean, it's a very dynamic process, and it's at some equilibrium and affected by changes in the conditions.
So would the biologist say that that contradicts the central dogma then? Because typically you think that all the information flows from genome to biochemistry and then to the environment, whereas here you have basically environment and biochemistry having feedback on genomic structure.
Well, I do see it that way. I'm not sure that I don't see that as a violation of the central dogma.
Yes, how strict you are about it.
Yeah. But I mean, I think the idea that, I like the phase idea, that there might be, that you might apply that kind of thinking to this problem, I think is an interesting idea.
So in bacterial communities, do you actually see something that looks like a phase transition in genome? Because I know like in cancer, you sometimes see these big rearrangements, which I would say may look like a phase transition, but I'm not sure if you can see that in bacteria.
That's a very interesting question. I think one reason I don't think I know the answer or anybody knows the answer is because of the undersampling of organisms that really succeed in nature. So we oversample things that we like to work with in our, I didn't tell you, but these cells are very hard to work with. And they're lacking so much of the regulation and that are found in cells that are easy to grow. So we undersampled them. So I think when there's more information about these genomes, I want to also add that the metabolic networks in these cells clearly show a lot of evidence of rearrangement, non-canonical rearrangements. Now, is that actually common? I mean, are what we consider canonical metabolic arrangements really non-canonical that have emerged from overrepresenting cells that are very plastic in their responses? That's a possibility. I don't think there's enough of these cells have been studied yet to, certainly more genomes are flooding in now because of single cell genome sequencing. So that's, but mostly they run through an annotation pipeline and there's some kind of fuzzy image of what they do.
I can address your question regarding the environment changing the genome structures. Like in cancer cells, there's plenty of evidence that under stress, microbes can express a mutator phenotype, and they can shuffle their genomes, trigger horizontal gene transfers, trigger transposons jumping around, increase the mutation rate in some cases. And so they have this inbuilt mechanism to try and generate variation to figure out a new solution for the new stress.
But it's interesting that we were talking about this, that if you decrease the size of your genome, you may have less genetic diversity to be able to do that. So there's a potential cost, right? So if you, I mean, I don't know the conditions under which you're your microbes decrease the size of their genome, but if they were to switch environments or go into a more unpredictable environment, they might actually pay a much bigger cost by having a smaller genome than if they had just turned off a bunch of genes. But it sounds like the nutrients are so limiting in these systems that cost is, at least in the short term, higher than anything else.
Well, I will mention that a number of these organisms are lacking a number of very nearly universal mutator genes. And there's debate about what that means. So I think it's clearly they're capable of recombination and pulling in DNA that from non-conspecifics, but really what their mechanisms of adaptation are at a genetic level, I don't think are very well understood. One of the reasons for that is, for the most part, there are no genetic systems for these cells.
I have a couple of questions, but I wanted to link back into Larry's question with Steve. So do you think Larry's question was really about the deeper aspect of how certain things are linked, like your example of the boxes? Are we dealing with box 1, 2, 3, or 4? That really we're looking at linked phenomena that go back into Christopher Siemens' catastrophe theory, where if we could spot things further back that are linked, we would stop going into a subduction zone and we would stop a catastrophe, rather than viewing things as chaos. And I'm interested because I think that's more of a predictive model.
If I understand your question, I think things are linked.
Yeah.
And that if we understood those linkages, we would be able to predict better.
Right, because catastrophe theory is a better example of being able to conceptualize that.
Maybe I'll just mention this. You know, when we started cloning genes out of the oceans, no one had any idea what they were going to see. And every single time you dig deeper into that system, you find more and more order and structure.
Right. I agree with you. That's why I think catastrophe theory is kind of poorly understood and poorly applied. Yeah. So, because it's a little bit in Greg's theory too, he talked about chaos. And for me, when I look at chaos, I also see catastrophe theory, which is, it's not totally chaotic. So.
I was using that term very much as George did in his follow-up, that it's a way to quantify sensitivity to initial conditions. Nothing more than that, really.
Yeah, that's interesting.
I don't really understand catastrophe theory. I know it had its heyday before I was on the scene.
And then I did have a question for you, Greg, which is, I think the climate work's really interesting. And could you talk a little bit to the application to water security and food security in your model, and if that works, presumably that work's being done?
So those are the impacts that everybody wants the answers to. Part of it involves going to scales that are finer than we can currently resolve with our global climate models on these long periods of time. So we're building an effort at the University of Washington right now to do regional climate modeling at high resolution to try and answer these sorts of questions. You need to be able to resolve the snow in the mountains to know what kind of runoff you're going to have into the reservoirs. There's a larger question. What you're talking about is, when are we going to have these decadal scale droughts that cover, say, the southwestern United States or affecting California like right now? And I think our ability to predict those is still not well known.
One more question.
Yeah, thanks. I found all the talks really personally stimulating, but I do have a job too, and this may not be something you can address, but I'd like to see. I have a task of trying to think strategies for response to unintended consequences of human action over the next 100 years. So if you look at tipping points in climate change or financial collapse in 2008 or antibiotic-resistant disease, you have places where human behavior has played a very clear role in playing what has been or could be tipping points. Are there things in the things that you've told me today that I might have missed that would suggest how your work informs what the human response to the structures and dynamics you're looking at might be. May not be answerable, but if you have anything to say, I'd be happy to hear it.
I'll take it.
Okay, you want to start?
I'll take a crack at it. I think the key is really uncertainty quantification, right? That even though we can't build this into our climate models, that there's going to be a financial calamity in 2023 that will lead to an economic slowdown and change the amount of carbon we emit into the atmosphere. We could do that stochastically in our climate models, and what it's going to do is it's going to increase the uncertainty in our projections. I think that's kind of the bottom line. As you add more of these degrees of freedom, it ultimately leads to more uncertainty. This complexification, it's that complexification stuff again.
So is human society's robustness in dealing with uncertainty that needs some development?
That seems like a reasonable proposition to me.
Yeah, so I didn't talk about ocean iron fertilization at all. I didn't say one word, and I'm proud of myself for avoiding that topic. But it's uncertainty that stops those plans. And I think that's a good example of, I think what we're doing is beginning to describe what things we need to understand, but we don't have that understanding yet. And when we have it, maybe we'll be able to do a better job of predicting just how good or how bad ocean iron fertilization would be. I think right now it's just very uncertain.
I think one of the big things is getting the public to recognize that uncertainty is a problem, right? I mean, I think insurance companies are maybe coming on board finally because they see major losses. But I saw a depressing headline yesterday, I think in the New Yorker, that people are only starting to recognize that climate change may be a problem because their yards are drying up, right? So it takes something for people to, recognize that there are issues that are going to bother them. But I think we all hit on the idea that uncertainty creates problems for lots of organisms in the terrestrial and the marine environment and experimental setups and what have you. And I think understanding uncertainty, like you said, in the stock markets, in the environment, in our daily lives is important. So, but people need to understand that. The general public needs to understand the importance of uncertainty in all of these processes.
I think, Greg, you're up front in that area, when it comes to dealing with uncertainty and then getting the public to try and recognize that. You're probably more at the coalface at this point because everyone's going to be looking at, wow, the weather it just changes year by year, but the predictions are starting to say, this is more catastrophic things coming forward, of what you were talking about, that tailing off.
I can't tell you the amount of hand-wringing there is in the weather prediction community about, we know so much about uncertainty, and it's not communicated to the public because there's this perception that the public can't handle it. They want yes, no answers, 1 zeros, black, white. They can't deal with the uncertainty. And I don't believe that at all. I mean, there's certainly a lot of people who run businesses. They either figure that out or they go out of business, right? Somebody outcompetes them. But anybody who plays Texas Hold'em or, the odds factor into our daily lives in so many different ways. I think we need to kind of tap into the public's awareness of that they are dealing with uncertainty all the time. When they hang that right-hand turn, they don't want to, run over somebody. They check and see, is there a cyclist back there? There's uncertainty in the world, and we need to live with it.
You'd be waiting pipe. Sorry.
I have very strong opinions about that because I work on earthquakes and wildfires too. So, more than climate, this is a situation.
Do you want to just use the mic? Sorry.
Where dealing with uncertainty is really part of the problem, and public perception of uncertainty and high variability. So climate, to me, is a really interesting example because sort of the happening of the tipping point is out in the future. And so you have to get the people to care about it before they experience it. Whereas in wildfires and earthquakes, you have high variability phenomena. Those large earthquakes do happen. They don't happen that often. Same with fires. And one of the lessons of that is that in terms of insurance often, public planning, policy, and things like that. It's sort of the point of view for many years has been to plan for the mean and not to take account of the fact that you're dealing in many of these cases with high variability, power law distributions, large events are inevitable and to be expected. So if you send all your resources to the small fires or the small earthquakes, you're not going to have any left for large fires. And if you go rebuild your houses at the urban wildland interface, they're going to keep burning down because we are to expect these large fires. So I think from a policy point of view, taking into account heavy-tailed variability in fluctuations in all kinds of things is sort of still a new point of view in a lot of these types of problems. And so I think that has to be something that people care about in these types of things. So.
One thing I can comment on is the importance of outreach and education. I think there's critical need for getting we call them lay people, but I think they're smart people. We need to give them the tools to understand how data are collected and analyzed and integrated and so on, and understand the concept of uncertainty. And at ISB, we have a concerted effort to bring current practices, scientific practices, not just content, to high schools and K through family science. I think that is a critical point that is missed in our education, where we're focusing on content rather than the process. I think students have no sense of failure. We reward them for success, but we should reward for failure. I mean, experiments are 1% success, 99% failure. I think there's a lot more we learn from failures, but unless you understand that process, I think you can do very little in terms of these complex concepts that we're trying to have them understand and appreciate.
I think another, like Southern California Earthquake Center, that's a huge part of what they do also is education, outreach, practice, involve the public, involve response agencies in cohesive actions that everybody knows about. And the other statement is that there's very little in terms of just resource allocation and spending. I've heard of statistics at one point, something like 99% of our resources are reactively spent as opposed to proactively spent. So any small things that we could do on the proactive side for many of these kinds of problems is our resource, like science, but also like outreach and all kinds of measures are to our benefit. And we're just so far away from responding on the front end of many of these problems that there's huge opportunity there.
Okay, one final question. We've been waiting so patiently.
Thanks, Steve. So Greg talked about choosing optimal sensor location and that sort of thing. So I wonder if that same question can be applied to choosing model systems. And so of all the things we can grow in the lab, how do you pick which organism to do these experiments on? And what effort should be directed towards isolating new things, like Steve has done, in order to address these type of tipping points in the lab?
So there's been a bunch of papers like in the last 2 or 3 weeks about this. There's 2 papers in Trends in Neuroscience, and there's a whole series of papers in eLife about model systems, right? And we need more model systems. How do you choose the model system? Some people would argue that with some of these technologies like RNA-seq and the ability to sequence genomes, you can do these things in non-model organisms, right? So I'm constrained by a lot what I can do with birds, but now we've sequenced the genome of the superb starling. We've annotated it. We've figured out how to get enough RNA out of blood that we can do RNA-seq, and so we can start to do lots of things that we couldn't do. We'll never have the tools that we have for yeast and some of these other things. But now we can do so much more, and I might argue that we shouldn't even call model organisms now. We can do so many things in non-model organisms that rather than developing more model organisms, just do everything in as many species as we can. Be one approach, because we can do things in the wild where we can study the ecology and the behavior in ways that you can't in captivity. So that would be one way I'd advocate it, but I'll leave it to the others.
Well, I can comment on that as well. I think I second Dustin's comment, but I'll temper it a little bit. And I will say one of the reasons we picked Halobacterium as an organism to start our systems biology inquiry was with the intent of developing methodology that could be applicable to any organism you could bring from the field based on the biology you wanted to explore. So it's a departure from way biologists have worked in a reductionist environment where you have a well-defined system with tools for probing specific networks or genes and so on, and then going to an organism that has just been brought into culture, or not even in culture, to understand how it functions. I think that's a complete departure, and I think systems biology has made that possible. But that being said, I think you do need to have the iterative cycle where you will find patterns in the environment or in your non-model. I still think model organisms are important. You will see patterns that will require you to then probe the mechanism. And when you start probing mechanism, you'll have to have the tools to genetically manipulate and test the consequences of those manipulations on the behaviors that you're studying. And I think that iterative cycle is very critical to get down to some of the cause-effect mechanistic behaviors.
I couldn't agree more with what Nitin just said. You have to have the capacity to pursue an idea over scales and to really understand what's going on.
Yeah, I think one thing I'll also say is I think the importance of communities, microbial communities, is there's a resurgence there. It was understood in context of biofilms and so on, but now we can actually probe those. And the synthetic microbial communities people are able to put together is going to get more and more traction where you can begin to understand some of these behaviors across scales, which some behaviors, you say a third or more, maybe half of the genome encodes function we don't understand. And part of the reason is context. I think we look at context just environmentally, but I think there's context in the community sense as well. I think we need to incorporate that.
That's great. So with that fantastic last question, I think we'll break for lunch. One thing I do learn is never say never, so we're going to have lunch now anyway. So we'll be back at 1:15. Thank you.